Good evening, everyone. Welcome back to the today's session. And today's topic is homology modeling of proteins. And this topic is taken by Dr. Kamla Golla, ma'am. Before I hand over the session to ma'am, I request everyone to keep their mics mute and also use chat box for any communication. So wish you all happy learning. I request Kamla ma'am to take the session forward.
Thank you, Bhavani.
Yeah, thank you, ma'am.
Good evening, one and all. Vanta ma'am, shall I start or shall I wait another 5 minutes?
No, you can
No.
start, Kamala ma'am. They'll join.
No, right. So, good evening, one and all. Yes, 1 second. Being Yes, everyone able to see the screen? Yes, ma'am?
Yes, ma'am.
Okay, right. So last class, we just know with the different tools available in online and different servers. So today we just go to predict existing PDBs, how to view in the SPDB, as well as unknown FASTA sequences or unknown sequences, how to get a protein structure, and what parameters we should consider when we develop a structure of a protein. This is completely, we just carry forward with the work progress, right? Yes, ma'am. Shall I start the class?
Yes, ma'am.
Yesterday, what kind of tools we are using to get the PDB structure for a protein molecule? Swiss model, right? So before starting this, I just explain protein having 4 structures: primary, secondary, tertiary, quaternary structures, right? So 3D structure plays an important role to show number of functionalities and versatilities. Okay, so 3D structure of a protein plays an important role in the entire living system. It plays a very important role. That is the reason we just focus on the 3D structure of the protein. Right. So this model development and all the things happen with the online servers. Okay. Some Autodoc, so many things are there. We have to download it and we can develop independently. That is a different story. So now I just want to explain how to develop a structure. Already yesterday I explained the Swiss model, right? Yes, ma'am. Anybody practice it? Anybody try to get a structure? So now I want to use this workspace. 1 second. Give me 1 second. Do just go with this. Okay, so my screen is visible, right? So I just go to PDB database and randomly I just go with the some tyrosine phosphate enzyme, right? So tyrosine phosphate enzyme, right? So here display the file means you have to display the FASTA sequence as well as see display the PDB format. Okay, so please notice this is the PDB format. Okay, later I will explain, right? So here download the files. So FASTA sequence as well as PDB formats. Are you notice the things? Please, ma. So if you are searching any protein, right? So this is my protein. Today, my work is along with this protein. So I just want to copy this and I want to paste in OneNote. 1 second, I'll show it.
Ola, are you sharing the screen? I
Yes,
think
ma'am.
can't see anything. Is it visible to others? Only I am not able to see.
1 second, ma'am. Now, Padma ma'am?
Oh, now it's there.
Oh, I just switched to see Google and Notepad and PPT. So a little bit of time gap is there. So at that time we can't visualize the things. So today my work protein is, now I take a protein from PDB database and paste in the Notepad. Okay. So I just take a protein example, right? For today's work, right? So I just get the PDB ID. This is PDB ID. Right? So this is the name of the protein, tyrosine phosphatase, right? So now I just download the FASTA sequence as well as C. Okay. So now I just want to download the FASTA sequence as well as PDB format, right? Okay. Now by using PDB database, we can get the PDB structure as well as FASTA sequence. Okay. This is already explained in the previous class, right? Okay. Now Swiss model, we have to develop a model for the protein structure. Now we just go with the workstation of Swiss model. Yesterday I explained already this one. Go to the internet and just open a tab. Swiss model. Okay. Swiss model workspace. Now I just download the PDB as well as, so now I just upload the sequence. Get this sequence. I just want to copy the pasta format in this and paste it. Okay, so give any name, 1 or 2, right? It is optional. So build model. Yesterday also we did this same thing. Same thing. I just repeat the same thing. So unknown sequences we can paste in pasta format in Swiss model. We can get the 3D structure of respective sequence. Is it clear, ma? We can get the 3D structure of unknown sequence, but I am pasting the known sequence which is retrieved from the PDB database. Is it clear? Are you able to follow this one? Yes, ma'am. Are you able to follow this one? Yesterday also I just repeat the same thing. But yesterday I just paste the random pasta format. But now I just start with PDB database, retrieving one FASTA sequence, and I just paste the same sequence in the Swiss model and generate the 3D structure. Is it clear? So this is already existing structure retrieving by Swiss model. Are you able to get the point? Yes? Yes. Remaining people, respond in chat box, please, quickly. Are you able to take one PDB from NCBI? Okay. Just download the FASTA sequence, paste in Swiss model, and generate the 3D structure. Is it clear? Please download the, now after generating of this sequence or PDB structure, okay, after generating of 3 PDB structure, please go to the download files. Okay, so here PDB format is there. Please click that one. Okay, so this one is stored as, one second, PDB format in your respective downloads. Okay, so now I just showing that. Okay, so model 2, one second, I just want to share the screen. Okay, downloads. I just download the model, what I exactly retrieving from the Swiss model, right? So I just rename the model, VAC 1. Okay, so this is the model exactly getting after pasting of your FASTA sequence. Yes? Are you able to follow this one? Yes, ma'am. Are you able to follow this one? See, I just paste the FASTA format in Swiss model and generate the PDB. Okay, after generating of this PDB, I just download it and rename it. Is it clear? Yesterday, this work is completed almost. Yes, ma'am.
Excuse me, ma'am.
Yes, ma'am. Tell me.
Can you show once again how to download the model?
Yes, definitely. So I want to share the screen. Okay, see. Are you able to see the screen, ma? Swiss model?
Yes, ma'am.
Right. So see, ma, download files. Are you able to see the download files? I'm just moving the
Yes, ma'am.
cursor. So in download files, you have to click it. Then one more extension box is released. So in that PDB format, is it clear?
Yes, ma'am.
So
Got
you
it, ma'am.
click the button, you get save the PDB format. Okay, right? Everybody able to download your model using Swiss model? Yes or no? Then I move on to the next level. Yes?
Yes, ma'am.
Right. Okay. Now, one second. Now I just want to share the keep, right? Okay. So everyone expertise to get a structure using FASTA sequence. Okay, whether it is PDB or unknown sequence, any sequence, you just give a FASTA format, then it is automatically give the PDB structures. Most probable structures are displayed. one or five or multiple, most commonly 5 structures are developed in this scenario. Is it clear, ma? So next. Okay. So here we are doing this work with known sequence, right? Okay. So you please use unknown sequences. That means how to do the unknown sequences. Take any of protein according to your interest. Okay. Unknown sequence, that is most important. Today work is completely focused on unknown sequence to develop the 3D structure. Unknown sequence of a protein used in FASTA format and develop 3D structures which are available in online. Okay, you just use the online servers to develop a 3D structure of unknown amino acid sequence. Is it clear, ma? Is it clear?
Yes, ma'am.
Right. So first task we just completed to get a PDB and FASTA format, whatever existing in PDB database, we just retrieve it and paste it in Swiss model and we get the structure. That is one task. Now, just move on to next level, right? So here what to do we? 1 second. So according to your convenience, you have to take any amino acid sequence. Any amino acid sequence. Any amino acid sequence. Okay, so I'm just going to the workspace. See, are you able to see the screen? Right? I'm using. So now I just want to take PDB database. Okay, I will tell you. Right. So PDB database. Okay. Right. Anyone tell me any protein, ma? According to your convenience. Tell me one protein name. Anyone? Can tell me any protein. Students.
I have
Please.
typed, it, Kamala, insulin P53.
Okay, right, ma'am. I am unable to see the chart. That is the reason.
Yeah, I have typed it as insulin.
Insulin. Okay. So I just always preferred insulin. By means it is a shorter one, right? So I just go with the insulin. Small, shortest protein, right? So see, 3-dimensional structure of insulin related binary secondary peptides. Okay. So I just take the first one only, right? So as usual, I just download the FASTA sequence and as well as PDB structure. Ma'am, you said it is an unknown structure, right? I will tell you. Okay. So now I just go with the BLAST, BLAST, BLAST. Okay. So it is a protein BLAST. Okay. So previously I started with a FASTA sequence. I just go with the FASTA sequence. See, are you able to see the screen, ma'am?
Yes, ma'am.
Okay, so this is the FASTA sequence. Okay, I just copy this sequence and paste in the BLAST. Okay, and right, I just do the BLAST. Internal server error. What happened? Moving one. So go for a single So it is showing internal server error. Why means I just paste the 2 peptide chains. So single peptide chain blast is simpler than the multiple sequences, right? Okay, now I just do the blast. So by doing blast, I just find out the similar sequences, what exactly present in the PDB database. Okay, so what kind of work we are carry forward? Please mention step by step, right? First one is known structure, known structure. What? Known structure PDB development with the help of Swiss model workstation. Right. Now I am explaining unknown structure 3D, unknown sequence 3D structure development. Right. So here also I am taking known sequence only. Why I am saying unknown structure? Listen guys. First I took one sequence, then next one I just do the blast. Going to happen. Okay. Line blast. First, I identify the sequence. Next, I'm doing blast to search the similar sequences. And then I just move on to the multiple sequence alignment of the 10 similar sequences, right? So I just go with the multiple sequence alignment and modify the respective amino acids. Remember, so once I modify the things, amino acid sequences with the suitable amino acids, that is a new FASTA sequence. So using that new FASTA sequence, okay, I'm using that new FASTA sequence to develop a structure with the help of Swiss model workspace. Is it clear, ma? So now I am getting 100 similar sequences. I just want only 10, right? So I just select the 10 and I'm getting the 10 sequences, right? So see, multiple sequence alignment. So without doing anything, without downloading of FASTA sequences, you just directly go with the multiple sequence alignment. Are you able to follow this one?
Yes, ma'am.
Right. Okay. Okay. So now I'm getting schema. See, I just compile the 10 sequences. In this is a common segment present in all 10 sequences. Remaining things are vary from sequence to sequence. Is it clear? Okay. Is it clear, ma? Is it clear?
Yes, ma'am.
Yes, sir.
Right. So this sequence. So and okay. So now previously I am taking around 25 amino acids, right? Now I just pick up the 80 amino acids containing precursor molecule. Is it clear? 82 amino acids, right? Okay. It is not giving me. One second. See, this one precursor, not gene. I want protein molecule, right? So whenever we are going with straight away with the blast continuation of multiple sequence alignment, this sequence is more conserved sequence, right? It is constantly present in the all kinds of organisms which are showing 100% similarities, right? Bombyx mori, this sequence is extracted from Bombyx mori. So same similarities are there in different organisms, Bombyx mori and different species, right? So now I want to check. This is my query sequence. Okay. Right. This is not giving error. Is that FASTA sequence? Okay. If I'm not getting FASTA sequence, I will do one thing. Okay. Right. So this is the sequence. Taking notepad. One second. One second. Sharing. And again, I just take this sharing part and I just use the OneNote. Okay. So now I'm taking one more sequence, right? So here, are you able to see the single letter amino acid code, ma? Everyone, right? I want to copy the same thing and I just modified according to my wish. That's all. Cysteine is there. At that place, I am putting methionine. Cysteine is there. At that place, I am keeping methionine, right? It is not suitable to generate a structure. So D means what is meant by D? Aspartic acid. Aspartic acid is replaced by so N aspergine. Okay, are you able to see the screen? Yes, ma'am.
Yes, ma'am.
According to my wish, I just want to change few amino acids. Okay, so now I just use this sequence to generate a PDB in Swiss model workspace. Is it clear? Now I am copying this sequence. I want to paste in this model workspace. Start modeling, right? Okay, I'm pasting the sequence, right? Are you getting clarity? Yes, ma'am. Still you have any doubts? Please see, I just modify the certain amino acids and keep it in FASTA format. Now I just build the model. That means unknown sequences also used to generate 3D structure of a protein. Is it clear, ma? Swiss model workspace is helpful to generate a unknown sequence 3D structure, unknown amino acid sequence 3D structure. We can develop by using this platform. Is it clear? But it will take little bit of time. Are you able to follow today class, ma?
Yes, ma'am.
Right. 1 second. So Swiss model is providing a space area to generate new models of a new sequences as well as desired consent. So whatever amino acid sequence we want, that kind of amino acid sequence, just paste in FASTA format. Okay, then you can able to generate the 3D structures. So first thing is existing PDBs, check for structure development in Swiss model workstation, right? Second one is a new or novel polypeptide chain amino acid sequence we pasted in the FASTA format and generating new model. Is it clear? Are you able to follow this one?
Yes, ma'am.
Only Antima is there. Reminding people,
Yes.
please respond. Put a message in the chat box. Are you able to? Are you able to generate 3D structure of a protein? Thank you guys. Right? So previously I'm using an existing FASTA format to generate PDB structure, right? It is easy. So immediately it is showing the 3D structure. So now I just modify the entire sequences, right? So 2 or 3 amino acids I changed. I just placed repeated sequences, right? So it is a little bit of lengthy one. So it will take little bit of time, right? Again, we just move on to the next. Okay. So till generation time, I just explained this. Okay. So in this way, Swiss model workspace is used to generate new 3D structures of a proteins. Is it clear, ma'am? Is it clear?
Yes, ma'am.
Right. Next one.
Yes, ma'am.
Okay. Next one. Okay. I see protein structure and prediction online tool. I taser TASSER. But here there is an issue with this one. Why means we required institutional mail ID. Otherwise, we are unable to do work with this server. Remember, guys, it is also one of the excellent online tool. But here the requirement is it is not free for the normal people. It is a ZenLab product. But when we are accessing this server, we have an institutional email ID. Without institutional email ID, we can't predict or generate 3D models using this server. Okay, this is initially Jang server. Later on, it is a server. Is it clear?
Yes,
Is it
ma'am.
clear? Okay, right. So this server, it is also providing okay. This server also providing so many online services, right? What cofactor? If you are generating a 3D structure, complete detailing explanation part is given this by using this server. Okay. Structure-based function annotations are also predicted by using this server. Only one requirement is institutional email ID. That is a problem with this. Okay, normal email, Yahoo's email IDs, they are not work with this server, right? Okay, next one, protein structure prediction server 2.0 or 2.0, right? So it is another server, right? Okay, it is giving a comprehensive protein structure prediction and annotations. Remember, secondary structures plays a major role. So whether your protein is stable or it is little bit unstable with the temperature and pH, that everything is clearly explained with this server. Okay, you just try it once. Protein structure prediction server, right? It is not straight away giving the 3D structure, it is giving the secondary structure and which one is stabilized with the different non-covalent interactions or whatever, and whether the protein is transmembrane protein or it is present in cytosolic version. Okay, everything it is explained with the depository, right? Already depositories are there, right? It is comparing with all the depository proteins and give a complete expression of your query protein. Is it clear? Is it clear?
Yes, ma'am.
One second. I just
Ma'am,
go.
please explain the last part where you said
One
you'll
second.
compare structures with other.
One second. One second. We will try now. We just use this online tool, right? I just go with the explanation, but 1 second. Give me 1 minute time. Okay. Now I am sharing the windows, right? Are you able to see the screen?
Yes, ma'am.
Yes, ma'am.
Right. So now I just want to, yes. So PROTEUS2.ca. You just please use this one. You just straight away enter into the server, right? Now please paste the sequence. Please paste the sequence. Any FASTA sequence. Still we just use so many FASTA sequences, right? So you can remove it. You can go with the sequence Bombix Mori and I'm pasting this sequence here. Okay. It is a eukaryotic organism. Organism is required only single polypeptide chain is expected. Right? Okay. Right. So view results in the web browser. Okay. Okay. So I'm not using any PDB structures. So this is the thing. I just paste only single polypeptide chain and submit. Okay. So previously we just submit one sequence, right? Okay. Previously we submit one sequence, right? Repeatedly I just paste and I just use one unknown sequence, right? So we got that PDB structure also, right? Is it clear, ma? Are you able to follow this one? One second. First, I want to complete this one and then there is a problem in running. Okay, so here there is a problem. I just want to check with this. What is going to be happening? So tyrosine phosphate. Okay, I'll submit this one. You take any sequence of FASTA format and this is going to be happen, right? Seema, initially whatever the jobs running in black color, once the job is completed, it is going to be converted into green color, right? So what kind of things are displayed by this server? Seema, this much information is displayed by this server, right? So here, this server just give the PDB homologous. That means similar kind of PDBs are displayed here and E value, most probable numbers, right? So number of coverages, overall alpha helix percentage, beta sheets, coils, signal peptides contents and membrane content. So clearly explain whether the protein is either imbibed into the membrane or not, right? How many helical structures are there? Beta sheet is there. Everything is explained, and it is also explained the similar homology or similar sequences aligned in a graphical alignment model. Is it clear? Are you able to follow this one? Protus 2. It is an online server, structural prediction server. It is giving complete detailing of the suitable PDB numbers or PDB IDs with the query sequence, and so length of the prediction with respect to suitability as well as overall coverage value, then alpha, beta helix, and it is also giving the homologous PDB sequences with the query. Okay, it is simply related to multiple sequence alignment, right? And complete secondary structures. C is chain, right? H is helix. Are you able to follow this one? It is giving a complete secondary structure detailing. Is it clear?
Yes, ma'am.
Yes, ma'am.
Right. Next, this polypeptide chain having any signal peptide, that information is also given by this server, right? Any transmembrane region is present. If transmembrane region is there, that protein must be. If transmembrane region is there, the protein must be. Any one of you. This, our current protein is not having transmembrane region, right? If transmembrane is there for a protein, what is the conclusion? If transmembrane region is there for a protein, then what is your conclusion? I'm asking. Anyone?
Is it a transmembranic protein, ma'am?
Transmembrane region, if yes, correct. Okay, transmembrane region is there. That means it is imbibed into the membrane. That means integral membrane protein. Okay, it is somewhere is contact with the membrane. Is it clear? Transmembrane region is there means the protein most probably intact with the membrane, either plasma membrane or endoplasmic membrane, endoplasmic reticulum membrane, or Golgi membranes, or nuclear membrane, whatever it may be. But it is anyway contact with the membrane. But our query sequence is not at all having transmembrane regions. Is it clear? That means it is freely rotating component present in either cytoplasm or nucleoplasm. Is it clear? Is it clear?
Yes, ma'am.
Right. So next, okay. So in this way, whatever the secondary structures, possible secondary structures, okay, it is giving complete details with the query sequence and multiple sequence alignments, right? So whatever sequence showing homology, right? So that is also explained with this query sequence, right? Okay, next one, blast alignment and sequence used by this, right? That is also explained in the box. Is it clear, ma'am? It is not directly giving the 3D structure. It is completely explained structural related elements. Okay, are you able to follow this one?
Yes, ma'am.
Right. 1 second, I just go back to this. Okay, so Seema, here if you required 3D structure, run the homology modeling. You have to press it and submit. It will take little bit of time, right? So 3D structure, 2D, comprehensive. This server is able to provided 2D and 3D structure predictions. Remember, if you opt 2D structure prediction, it is giving secondary structures and related information. If you want 3D structures, it is also providing 3D structures, right? We have to download it, right? It will take little bit of time. Okay. Immediately 2D information is displayed and 3D information little bit time taking process. That is the reason I am not explaining much. Okay. Are you able to follow this one? Somebody asked a question regarding Proteus structure prediction server. Are you getting clarity on this?
Yes, ma'am.
Right. So everybody able to follow this one?
Yes, ma'am.
Only one person is. Okay, thank you, thank you, thank you. Right. So I just move on to the next level of presentation. Right?
Ma'am,
Yes, ma'am. Yes.
ma'am, why are we not using servers like AlphaFold because they also predict the 3D structure of protein?
Definitely, ma'am. Well, yes, we can use it. But the thing is, see, we are directly taking the FASTA sequences which are already deposited in the PDB database, right? So this server, small gaps or small slashes, if did any small mistake, the server won't give the PDB structure. Remember, accuracy is compulsory required. Okay, so this is an online platform, right? So I'm just doing paste, copy and sharing and all, right? If any small mistake happen, I won't get the 3D structure. So these mistakes are nullified in a big platform or big workspace. That is Swiss model, right? I will explain. Even though you just work with this, we can get the 3D structures, but it is a slow process. Okay, in the short span of time, I can't produce that 3D structure by using those links. That is the reason how many servers are available, we are listing out and we are giving to you, right? Please try. You can try and you can get the PDB structure. 3D structure we can get by using any server. Okay, na? Right? Still anyone having any doubts?
No, ma'am.
Right? So using, remember, Swiss model server workspace, right? We can get the old ones or depository PDB structures with a novel approach, right? And unknown sequence, using unknown sequence, we can develop the PDB structures. Is it clear? Next level is Proteus structure prediction. This server is happily providing 2D and 3D structures of a FASTA sequence. Here thing is you have to properly paste the FASTA sequence. If any mistake happen, it won't give the structures. Okay, so the next one is SEMA, DNAstar.com. Okay, it is another workspace. By using this workspace also, we can get the structure of the protein. Okay, basically, I just want to use this. Second. Okay, I just want to share this. So you can use this link as such. Directly we can use. One second. One second, I just want to paste this one. Same thing, I just want to paste in the chat box also. Okay. So, SEMA, protein structure prediction. This server providing a protein structure prediction server, okay, our workspace, and we also predict the folding strategies. Okay, so this is also providing a guide. Okay, workspace, prediction, accuracy, folding, and supporting information. So everything is provided in this server, right? Okay, just use this guide. We can use how to go with the submission and all. See, workflow is available, right? So protein analysis, protein docking, everything is possible. Just take any example and you just go ahead with this server. But it is very slow, ma. Just it will take so much of time. So, but this platform is useful for antibody modeling, right? Multiple sequence alignment, stimulations, everything, primer designing, phylogenetic analysis, okay, protein docking, protein sequence analysis, protein stability prediction, structural alignment, structural analysis, protein structure predictions. Is it clear, ma? It is one more online server. We can use so many analysis related to proteins. Okay? Are you able to follow this one?
Yes, ma'am.
Sorry?
Yes, ma'am.
Okay, na? Right. You just try any of things. See, step 1, secondary structure assessment. Already I explained the secondary structures. Right. So next, visualization of 3D structures by using AI. Right. So multimer alignments. Finally, we get the 3D structures. Any tool used for either secondary structure expression or 3D structure expression. Right. But further works you required 3D structure. So 3D structure of a protein, usually we just download it in PDB files. Please remember PDB files. Okay, now, right? Yes, ma'am.
Yes, ma'am.
Okay, now I just move on to next. Okay, right. Right, ma'am. So DNA star is one more software or server available in the internet. We can check the primers, we can check the protein structure annotations and multiple sequence alignment. Okay, 3D structure of the protein. Okay, right. Next, this is the beautiful platform available in the internet. X5C. Okay, it is one of the excellent platform, right? Homology modeling, multiple sequence alignment, secondary structures, primers. If what kind of bioinformatic work you required, we can do with this platform. EXPASY. It is developed by Swiss Institute of Bioinformatics. Without knowing of any previous knowledge, with the basic knowledge regarding proteins or regarding nucleic acid sequence, okay, we can do lot of work with this browser. Is it clear, ma'am?
Yes, ma'am.
Stop sharing. I just go with the net. Okay, now I just using XYZ. EX. Right. SIMA, Swiss SIB, Swiss Institute of Bioinformatics, right? Okay. So now I just want to click this one, right? SIMA. See, genes, genomics, metagenomics, transcriptomics, proteins, proteomics, evolution, phylogeny, drug designing, structural biology. Everything is possible with this server, with this link. Okay. So without downloading anything, you can do your preliminary works with the help of this, right? It is also providing a learning resources, right? You just go with the resource available in this. We can do online workshops with this also, right? So now my concern is proteins and proteomics, right? So this one is already we are using Swiss model, right? Quality estimation. See, protein quality model quality estimation is measured with the help of Q-mean. Remember, guys, Q-mean values explain the protein quality of the respective developed structure of a protein. Is it clear? So once we developed a protein with Swiss model or any online server, right? Download the PDB structure and whether it is the quality of the protein or whatever the developed 3D structure quality, we can measure with the different tools which are available in this workspace, right? Is it clear?
Yes, ma'am.
Only Antima is there. Huh? Okay, right. So another one. PDB sum analysis. Now I just move on to, so I just want to explain the XYZ. See, I just go back to, okay. So Uniport, Swissport, everything. Swiss doc is also feasible with the ligand and protein molecule, right? So everything is feasible with this workstation. Okay, are you getting clarity? Swiss model workspace is there. It is clearly this one, what exactly we are using for developing of structure. Is it clear?
Yes, ma'am.
Right. So this is very good bioinformatic tools hub, right? So we using any kind of tool and get the results. Okay. So one more important thing, right? So PDB sum. PDB sum. Okay. PDB sum means whatever PDB. So the quality of PDB structure is explained with the help of Ramachandran plot, okay, and progression of the 3D structure, secondary structures, how it is interact with the different groups which are existing in the database. So that kind of entire information is given by this PDB sum. Okay, is it clear, ma? So now I just use the PDB sum generate. Okay, PDB sum generate is a workspace, right? So in this SEMA file upload in the PDB sum phase. See, choose files, right? Choose files, right? I'm just uploading one. Okay, here email address is mandatory, right? Upload. Okay. Okay, so this is the result page. Remember, I already submitted one sequence, okay, that is transferase. So the result already sent to my email, right? So this PDB sum, whatever submitted here, we can get the results on this space, but waiting time is 5 to 10 minutes or 15 minutes, right? But I just keep the email ID, right? Whenever our job is completed, they directly send the information through to your emails. Okay. So previously I submitted transferase enzyme. So I got this results. Are you able to see the screen, ma?
Yes, ma'am.
Right. So here, pro-check analysis, SEMA. Pro-check analysis. This is Ramachandran plot. Remember. So our sequence having n number of amino acids, right? So which amino acid present in which area? So most probable acceptable regions, not probable regions, right? See, most favored regions. Seema, are you able to see the results? Pro-check statistics, Ramachandran statistics, right? So most favored regions, how many residues are there? 277. So our total number of residues is 344. 344 amino acids sequence is submitted to the PDB sum, right? So out of this, 277 are present in most favored regions. Okay. Additional allowed regions. Ramachandran plot. Remember, brown color and red color, right? So red color is most favored region. Okay. Favored region means that region containing amino acids not alter the structural features of the respective protein. Allowed regions and see, first one is favored region, followed by allowed region. Okay. See, allowed region is of two versions. Additional allowed region, generously allowed region, disallowed regions. Is it clear? So, Ramachandran plot explain the amino acids which are present in more favorable regions and disallowed regions. Remember, if any structure developed by you, right? See, disallowed region amino acids should be 0. Then your structure is perfect. If you develop any structure of a protein according to your convenience, according to your interest, right? Disallowed region should be 0. If it is reaching 0.1 or 1 or 2, right? The stability of the protein may alter. That is the important point I want to mention here. Are you getting clarity? Ramachandran plot give a complete information about amino acids present in the protein, right? So most of the proteins are present in most favorable regions. Okay, some of them are present in additional allowed regions and generous allowed regions, right? But disallowed regions, it is strictly prohibited. The amino acids should not be stay in the disallowed region. What is the importance of this thing? Disallowed region containing any amino acid, okay, that amino acid destruct the stability of the protein. That is the most important point. Is it clear? Yes, ma'am. Yes. Okay, thank you. Right. So in this way, we can get the Ramachandran analysis with ProCheck. Okay, ProCheck summary. Right. Just go back to next. One second. So content, right? Okay. So now, see, it is having 344 amino acid residues, right? Already I explained. So previously we see the secondary structure, helical structure, beta plated. See, ma, now we can see the entire structure with the secondary explanation part. So this arrow mark is corresponding to arrow marks corresponding to beta chains. Are you able to see? See this red color expression is showing the loop. Okay, so beta sheets as well as alpha coiling. Yes, secondary structures are completely explained. See, helicals labeled as H1 and H2 and stand by their sheets A and B. Is it clear? Beta represent the beta turns, gamma turns. See, red color one, beta hairpin. Is it clear, ma? So the complete explanation part of the structure is expressed either alpha helix or beta turns, okay, gamma turns, and everything is expressed here. So, and one more important point is this protein having any ligand molecules that is also expressed in the form of ligands. And this protein may have an ability to interact with the DNA and RNA. That is also explained here. And blue color represents it is intact with the metal ions. So the complete structure evolution, secondary structure evolution is carried forward by PDB-SUM. Is it clear?
Yes, ma'am.
Yes, ma'am.
Right. So, yes, ma'am. Yes.
Can you explain once again disallowed region?
Sorry. Disallowed
Can you
regions.
explain as much?
Right. 1 second. So I want to go with the pro check. 1 second. Yes. Disallowed regions. Yellow color. See, ma? Are you able to see the yellow color region? Light yellow color region. Okay. Usually Ramachandran plot is explain the pi psi values of a amino acid present in the polypeptide chain. It is in detail with the Ramachandran plot. I'm not explaining that much. Here, SEMA, yellow color, light, pale yellow color region is there. Are you able to recognize that pale yellow color? Yes, ma? Are you able to see? See,
Yes, ma'am.
dark yellow color, light yellow color, right? Is there any amino acid is there in this yellow color region? Light yellow color region. See, I will show it to you. See, this blue color is representing the respective amino acids. See dots, blue dots are there, right? Any blue dot is there in that yellow color region?
No, ma'am.
Right? So that is the reason disallowed region containing amino acid residues 0. Is it clear? Yes, ma'am. Are you able to follow this one? Yes. Yes, please.
If it is one, ma'am.
If it is one, see, some proteins having one, disallowed regions may reach us to 10 also, but it proportionally express the destabilization of the protein structure. That is the most important. If it is hydrophobic protein, one residue of hydrophilic residue is there, then what happened? The stability of the protein is altered, right? Even one amino acid plays a major role. One amino acid change causes mutations, right? Sickle cell anemia, right? Everybody know this.
Yes, ma'am.
Single sixth place amino acid replacement causes sickle cell anemia, right? One disallowed region containing single amino acid, the protein stability is altered. That is the reason I want to explain this. Are you able to follow this one, ma?
Can we know about the amino acid, ma'am?
No. See, what amino acids, for example, which every amino acid try to present in the allowed regions, right? So these are pi psi values. That means peptide bond, CONH bond associated with alpha carbon of the amino acid, right? So orientation. So the 3D structure may project that amino acid is pointed in the disallowed region and allowed region. Are you getting the point? Every amino acid independently, we won't plot the Ramachandran plot. Whenever the Ramachandran plot is one of the excellent representation of protein interactions, right? Okay, so whatever the amino acids, pi psi values are different with the peptide bond, right? So maximum protein when observed in the folding manner, right? Most of them amino acids are coming to the favored regions or allowed regions. Then only the protein stability is retained, right? If not, disallowed regions, some cases, one or two amino acids may occupy this disallowed region, right? Basing on the pi psi values. Right. So pi psi values may project that amino acid into the disallowed region. Is it clear? Is it clear?
డిసోల్డ్ రీజియన్ లో ఉన్న ఒకవేళ ఇన్ కేస్ అమైనో యాసిడ్ ఉంటే దాని నేమ్ తెలుసుకోవచ్చా మ్యామ్ ఈ వెబ్సైట్ త్రూ?
Definitely. So Seema, I will explain. See, non-glycine, non-proline residues. So proline is there. If it is alpha helix, proline is there. Definitely Nick region is present. That is disallowed region, right? So if I'm taking a protein which is not having no glycine and non-glycine and non-proline residues, okay, but it is having certain 19 and 15, right? Okay, but here beta bends are beta pin hairpins that structures as allowed with this glycine and proline. There is no issue. So secondary structure basic aspect. Okay, we can get the complete version of the amino acids which are present in allowed region, which are present in disallowed region. Certain proteins definitely having disallowed regions, amino acids, right? This yellow color schema, you just submit any kind of protein, PDB structure, right? Then you got dots in the light yellow color region. Okay, automatically it is disallowed region. Okay, that expression is explained in this statistical data. Is it clear? Yes, ma'am. Are you able to follow this one? One second. Are you able to follow this one, ma'am? Please respond in the chat box. Yes, please respond. Are you able to follow this one?
Yes,
Yes.
ma'am.
Okay. So what is the use? Why, ma'am, you are saying so many things? Right. My concern is, okay, here my concern is you people able to take a new sequence according to your wish, your desire, and develop 3D structure by using Swiss model workspace and check the parameters. If you are really getting good 3D structures, by using this workshop, you can develop a 3D structure protein by your own. That is my major concern here. Okay, so PDB sum is giving complete information regarding protein stability. ProCheck PDB sum. Okay, it is one of the excellent online platform. Give the complete details of the submitted query PDB structure. Okay, so secondary structure and annotations. How many segments are literally intact with the DNA and RNA? Okay, recognition sites, active sites, everything is displayed in this PDB sum. Okay, is it clear, ma'am? So now we just see, we can download here as a PDF document, and this document can utilize furtherly in any paper publication or anywhere. Okay, is it clear, ma'am? Is it
Yes, ma'am.
clear? Okay, right. Anyone having any doubt up to this level? Thank you. Right. So are you able to follow this one? Any toughness is there? Till now, we are not buying any kind of software tools, or we are not installing anything, right? Yes. Yes. Thank you. Thank you. Thank you. Right. Okay. Right. Thank you for your right responses. Right. Now I just move on to the next. Okay. So PDB sum is one of the online tool. We generated whatever PDB, the structural stability concept, everything we can check with this PDB sum. Okay. Right. Now I just move on to the next one. I just go with the PPT. Okay, so EXPASY 1 excellent platform. We can go with this. Now, another one. It is a Swiss PDB viewer. Remember, Swiss PDB viewer. That means we can visualize the PDB structure in your system. It is a powerful tool to analyze and modeling protein structures. Remember, Swiss PDB viewer, otherwise SPDBV. Okay, you please type these words. Okay, in Google. Okay, in the Google, you just type the SPDBV. Okay, so please download the SPDBV. Please download the SPDBV in your system. How many people are working with systems? Please download the SPDBV. But according to your system configurations, if it is Microsoft Windows, you please download the respective. So, installation. Shall I go with the installation process? Yes, ma'am. Shall I explain this one, ma'am?
Yes, ma'am.
Okay, so SPDB. SPDBV. It is a Swiss PDB viewer. It is available in internet. I want to share that also. Okay, 1 second. Give me 1 second. Okay, right. Okay. So now SPDBV. See, SPDBV download. Swiss PDB viewer download, right? So here downloads are there, left side. Are you able to see this thing? Yes, ma'am. Or otherwise I will post this link in the chat box. Okay. So you can use this link to download SPDB. Okay, ma'am. So while you are pasting that link, you are getting this kind of page, right? Left side downloads as there, right? Just click the download. So just go through the paragraph. Yes, I agree on the terms of the license and would like to proceed with the download of Swiss PDB viewer, right? So here please go with according to your computer configuration, right? Everybody having Microsoft Windows, please download this one. Okay, please download this one. After downloading, okay, in your downloads, please check SPDB zip file, SPDBV underscore 4.1.10 underscore PC. Zip file is there in your download. Please check after downloading of your viewer, right? Everyone get the zip file, right? Please extract the zip file in the same portion. Please extract the same zip folder in the same area. Extract it. See, this is the extracted format. I just want to copy this one on the desktop. Okay. Are you able to see the screen? Now I just want to click that one. Are you able to see this, all the things in your extracted file? Please let me know.
Yes, ma'am.
Right. See, ma, this SPDV is there. Just click that one. Yes. Are you able to open this kind of? Yes. Viewer, one toolbar is opened. Yes or no?
Yes, ma'am.
Right. So now I just want to explain how to see the PDB structure of. See, now I just take the file. Are you able to see the screen? Okay. Open the PDB file.
Kamala
Right.
ma'am, you opened any PDB file?
Yes, ma'am.
Because in what we are able to see is the extracted file only we are able to see SPDV application 2204 KB. This is the sheet what we are able to see.
Okay, ma'am. 1 second. It is sharing, ma'am. Here I am opening this. Okay. Okay. One
Double
second.
click చేసి చూడండి.
చూస్తాను మ్యామ్, ఒక్క నిమిషం. అది స్క్రీన్ షేర్ అవుతుంది. Yes, are you able to see the screen? Anta ma'am?
Yes, ma'am.
Okay. So we can download your PDB structures and view using this viewer. Are you getting clarity, ma'am? So SPDB viewer, it is a viewer software we can use to analyze and see the structure of a protein without any installations or without any further support and all. We can see view your protein, right? Okay, we can clearly view your desired protein and we have to rotate it. So now this is the file. Okay, we can use to open the PDB files. Okay, so see, ma'am, you have to save the current layer as a PDB file. Again, we can store this PDB with whatever modifications required. We do all the modifications in this workspace and current layer is stored as a another PDB, modified PDB. That is the reason they are
Kamala ma'am?
explaining. Yes, ma'am.
Sorry to disturb. ఇది I think 2-3 times the Swiss PDB viewer is been opened. I think so. I'm seeing only one layer above the other layer, the PDB viewer sheet alone. Workspace or the protein, what you have uploaded, we are not able to see. Yeah. Now you can go to file and upload, ma'am. No, again, I'm finding all the overlaying of your Swiss PDB
Yes,
viewer.
yes. Too many times I just want to, I opened, I think. I just want to.
Now only one is visible. Maybe if you go to file and upload PDB, maybe in the workspace.
Okay, ma'am. Now we can see the. Are you able to see the PDB, ma'am?
No,
Sharing. Yes, ma'am.
yeah, now the workspace with protein is visible.
Yes, ma'am. Everyone
Yes,
able
ma'am,
to
we can, please carry on.
Yes, ma'am. Thank you, ma'am. Right. Everyone able to open PDB by using this SPDB viewer. Are you able to open your PDB files?
Yes, ma'am.
Yes, right? So now what is the importance of this workspace? And importance will be explained by the Espadma ma'am in the next proceeding class. I just want to explain how to see the protein, what are the amino acids are present in this, how to see the amino acids present in this structure, right? So SEMA, file, edit, select, build, tools, fit, display, color, Swiss model, right? In the Swiss model, one second. See, WIND, win, right? Win having toolbar and control panel, right? So use the control panel, right? Are you able to see the control panel?
Yes, ma'am.
Yes. So we just open the control panel, right? So this structure, what exactly displayed in the space containing amino acids are all are listed in the control panel. Is it clear? Yes, ma'am.
Yes, ma'am.
Right. So in this control panel, whatever PDB I uploaded, that name itself expressed on the right side of the control panel. Okay. So right side means according to your perspective. Please, is it left side or one corner? You please consider 3BQ1. That is a PDB ID of the protein, what exactly we displayed, right? So now visible box is there, right? So I'm clicking that box. See what happened? Complete vanishing of the structure. So now I just put a tick mark. Everything is visible. That means whatever the amino acids present here, they give a structure to the protein that should be visible or invisible. So visible option is also there, right? Okay. Next, can move on the opposite version, can move, cannot move, right? So once you have to click that when we can't move the structure, right? So here 3-letter code is present, right? So whether you want to see the amino acid, see, I just want to remove, that place is empty. See, please see. 1 second, I want to show it to you. Okay, right? Are you able to see the screen? See, now I am removing certain amino acids, side chains and main chain, right? So I just want to remove those things, empty the space, right? Are you able to see the screen? Yes or no?
Yes, ma'am.
So in that way, you can visualize the ligand molecule as well as protein molecule. Which one is the ligand molecule? Which one is the protein molecule? We clear-cutly differentiated by using this viewer. Is it clear? Anyone having any doubts? Yes, ma'am. Still, do you have any doubts?
Ma'am.
Okay. So SPD viewer is helpful for to visualize our 3D structure and what kind of ligand. See, another one, ribbon, right? So I just put the word in the ribbon space. What is going to be happened? We'll check. Seema, are you able to see the structures? So secondary structure is completely displayed. Are you getting clarity? Yes, ma'am. Yes. So now I just want to remove this ribbon. What is going to be happened? The secondary structure does a pair with the corresponding amino acids. Is it clear?
Yes, ma'am.
So whatever requirements or whatever modifications do you want to create in the polypeptide chain or a protein, everything is feasible in this PDB viewer. If you want to change the color also, right? I just want to give the red color, right? What is going to be happen? See, this polypeptide chain highlighted with the red color, right? I just want to see the structure also. I just go with the bright color, right? Are you able to see the modifications? Yes or no?
Yes, ma'am.
So if you want to remove the amino acids for this polypeptide chain or a protein, we can remove it also. So using this control panel, we are able to remove the amino acids, but we have to select properly, right? Delete. Okay, we just want to delete this. See, any modification is happened, that won't be displayed in this structure. So after removing of this, we have to save the current layer. So that layer do not containing these amino acids, right? This amino acid structure is not be displayed on this viewer. So we just save the layer without those many amino acids. That means if you not require that much of protein, that means lengthy protein structure. So you just remove the unknown amino acid sequence from a protein by using SPD viewer. Is it clear? Is it clear, ma?
Yes, ma'am.
Right. Okay. Everybody able to access this SPD viewer? Yes, ma. Okay. What is the advantage of this? Visualization option, chain display option, and again coloring option is there. Measurement, mutating residues. We just modify the residues. It is explained by Dr. S. Padma ma'am tomorrow. Right. Energy minimization also explained by Padma ma'am. Is it clear, ma'am? So it is an excellent platform. We can use and we can minimize our desired protein. Okay. Is it clear, ma'am? Are you able to follow this one?
Yes, ma'am.
Ma'am,
Yes, ma'am. Yes, please. Yes, please.
ma'am, can we also like superimpose two protein structures and see like where are they different
Yes.
using SPDBV?
Yes. See, ma'am, we have so many databases, right? So many proteins. But our protein is different, right? So we have to check the protein. Similar sequences are there. Or we are entirely different model is developed by you, whether it is suitable for our work, superimpositions, okay? It is following Ramachandran plot. For example, we are generating 3D structure of a protein, right? Unknown protein, right? It is not existing in the database, right? Good. It is not following the Ramachandran plot. That means the protein is not stable, right? It is not helpful for further any drug ligand interactions, drug protein interaction studies. Is it clear, ma? So at that time, we have to modify the protein which is having a more acceptable regions, right? So in that version also, we can utilize this SPDB viewer and minimize how much energy is minimized and everything is, we can check and we get the optimum and more suitable 3D structure of a protein. Is it clear? Is it clear, ma?
Yes, ma'am.
Right. Still, anyone having any doubts? So this is the overall approach. Yesterday and today, I just want to conclude my topic. So here, once protein sequence getting from different databases, right? So suitable sequence search, blast. So the similar sequences are aligned with the multiple sequence alignment tool, right? Then this sequence, we just go with the multiple sequence alignment. Okay, suitable amino acids are replaced according to their phylogenetic relation and all. Then I develop a PDB structure, homology modeling, and verify whether the developed structure having suitability with respect to secondary structures. Okay, secondary structures suitability, structural stability studies, right? Folding recognition, everything. Then finally, I concluded that this structure is highly stable and useful for further analysis or further docking studies or anything. Okay, so first one is unknown sequence or known sequence. We just check for suitable or similar sequences by using BLAST. Okay, then align them into multiple sequences, multiple sequence alignment, right? After getting of multiple sequence alignment and phylogenetic relationship, we just modify the amino acids. Okay, then we have to produce or we can develop a new 3D structure of a protein. So after developing of your protein, right, then move on to next level. If you after completion of development of 3D structure, whether it is following all needs, that means thermodynamically it is feasible, secondary structures having a stability, pro-check analysis, okay, folding capabilities, energy minimization, these are all are meeting with the designed 3D structured protein. Once we concluded that everything is passed by the 3D structure, what exactly developed by you, right? Then that is useful for further uses. Protein ligand interaction studies or protein drug interaction studies. Everything is possible after development of 3D structure of your unknown sequence. Is it clear? Is it clear, ma?
Yes, ma'am.
Right. That's all for this class. Thank you, guys. Right? Anyone having
Thank you,
any doubts? Vanita, ma'am?
Yes, Kamala ma'am.
Attendance post చేద్దామమ్మా.
Yes, Tanu. Before that, any questions, queries are there, please, you can get it done. One participant has said, I did it, ma'am. It was asking for an update, but was not
Sorry?
happening. Maybe you 1 minute, one time, one of the participant is having a problem in downloading the tool. Maybe you can update your system and then try to reload. Maybe it will be possible. Because in case if you want to do auto dock, we need this SPDB viewer. So then only we can do the docking. So make sure if you want to, then you have to check for your updates.
Thank you, Kamala ma'am, for your informative lecture. And thank you, everyone.
Padma ma'am?
Yes, Kamala.
So we can go with the attendance posting and all. No?
Yeah, we will go.
I'll complete my topic today.
Yeah.
Anyone
Any
have any
queries?
doubts? Yes, ma'am.
Any queries, any difficulty in taking the process forward? How many of you have really gone with the session and worked around with this source person? Can I know? I think there are around 42 people in this meeting, excluding us, 3 of us. So the remaining 39, how many of you have really worked through along with us?
Yes, ma'am. Please respond.
No participant has worked through. You can say yes or no, it's okay. You can type yes or no, it was okay.
Yes, students.
Swiss PDB viewer, it is a simple thing, will easily download unless you have some other tools installed on your software and is taking of installed and it is taking up too much space on your computer, or there is no enough disk space in your computer also. Sometimes it will not load. So if you are not using, if you are having any other tools on your computer which you are not regularly using, you may remove them for some time for this session till this session completes, and then later you can reinstall it back once you stop working with the PDB viewer. So if there is not enough disk space also, sometimes it will not run, or if there is no space on your laptop, or if it is little slower version, there may be a problem. So that's only one candidate. What about others?
The remaining people able to follow today's session, ma'am? Respond, please. Respond in the chat box.
Okay, so Prerna, Purvi, you have gone through, you have completed till the stage of PDB viewer. Okay, that's good. Because we will be giving some assignment based on this, whatever you have been explained in the class, some questions will be done. So you may have to work and then only you will find the answer. So unless you are really learning it, assignments maybe you may find it little difficult to find the answer of the assignment. So it's always better that you are spending 2 hours of your precious time. You must learn thoroughly and go back with enhanced knowledge. That is the main purpose of this session. So it would be good if everybody involves and does the assignment or does the exercises as along with the resource person. So the resource person can do the troubleshooting and help you out if case any problem is there. Okay, if no other questions, we may proceed for.
Ma'am, attendance పెట్టలే. పెట్టే సార్. Yes, ma'am. Please, students, just complete that attendance sheet today.
Yeah, we are sharing the notes and PDF for